In our book, Beating the Dementia Monster, we said that there seemed to be more than one variety of the disease we call Alzheimer's. Is that true? If so, how many varieties are there? Let's take a look. Alzheimer's disease is defined by the amyloid plaques around brain cells and tangles of collapsed microtubules inside dead cells. We discussed in Beating the Dementia Monster how amyloid plaques and defective proteins in microtubules apparently kill brain cells. In the form of the disease we call young onset or familial Alzheimer's disease, we see certain specific genes in play, and the symptoms appear when someone is in their 40s or 50s. This differs from the form of the disease we call old onset or sporadic Alzheimer's, where symptoms don't generally appear before the age of 65. Sporadic Alzheimer's, like mine, seems to be less influenced by genetic factors and more by lifestyle and environmental factors. And genetic influence appears to be from different genes than those involved with young onset Alzheimer's, perhaps more than 30 different genes. By the way, many researchers and doctors try to avoid the term early onset Alzheimer's because it confuses the age of the person with a stage of the disease. Is it early in the person's life, like with young onset disease? Or is it an early stage of the sporadic form of the disease, but occurring when the person is, say, 80 years old? Because of this, we avoid the term early onset in favor of young onset or familial. So we see here two varieties of Alzheimer's disease. But can the disease be divided more finely? Or is it possible that we actually have more than one disease that we're dealing with? In my opinion, we don't understand the disease well enough to say if there is more than one disease, but there is now evidence that we can discern at least 5 subtypes. That's important because it may be that experimental therapies that seem to fail only failed because they were tried on 1 or 2 specific subtypes of the disease without realizing the therapy might be more suitable for another subtype. Also, the varieties may progress differently, confusing expectations and treatments. Research on different subtypes was published in the journal Nature Aging. It was titled Cerebrospinal Fluid Proteomics in Patients with Alzheimer's Disease Reveals 5 Molecular Subtypes with Distinct Genetic Risk Profiles. This article is linked in the show notes below. Researchers, primarily from the Alzheimer's Disease Research Center in Amsterdam, studied the cerebrospinal fluid of 609 people, examining the proteins they found. Some study participants had normal cognition and no evidence of disease, that was the control group, while others had been diagnosed with varying stages of the disease. Based on this evidence, the researchers believe they discerned 5 different subtypes of the disease based on which proteins they encountered. One thing that struck me right off the bat about these varieties was that subjects with one subtype went from onset of dementia to death in an average of 5.6 years, while another subtype had an average survival time of 8.9 years. That's a distinction. Alzheimer's disease is the 3rd leading cause of death in my state of Washington and the 6th leading cause of death in the United States. And, as we said in Beating the Dementia Monster, when I was 66, a researcher told me I was on track to be dead by the time I was 75. That made sense because at the time I was still in the middle of the mild cognitive impairment stage of the disease, and so was still a few years out from actual dementia. But he also indicated that if I would just keep up my recent lifestyle changes, I could extend that to 85. And that seems to be what is happening since I am now 75 and I'm not dead. But what are these 5 subtypes? Well, if you're not a doctor or a physiologist, they can be a bit hard to understand, or maybe a lot hard to understand. So we won't try to break them all down here. But one subtype significantly weakens the blood-brain barrier, allowing trash to enter the brain from the blood. As we discussed in Beating the Dementia Monster and in other posts on our blog, the blood-brain barrier prevents larger molecules, especially waste and pathogens, from passing from the blood into the brain. But this isolates the brain from the body's general immune system, and so the brain must have its own immune system. This system uses cells we call microglia. Apparently, with this subtype, the blood-brain barrier is weakened and a variety of unwanted proteins enter the brain and begin to cause damage. In Beating the Dementia Monster, we said that problems with microglia could play a central role in the development of the disease. Well, in another of these subtypes, the ability of microglia to protect the brain is impaired. One thing the microglia are supposed to do is glom onto the amyloid plaques and stop them from damaging cells. But in the variety here, the microglia are weakened and their protective function is seriously diminished. As we always hear, the authors of the study say more research is required. And it's clear that a better understanding of these subtypes is important. That will better inform research into treatments, since one treatment might better address subtype number 1 than subtype number 2. Also, some trials may have mistakenly found a treatment ineffective simply because it was tested on the wrong subtype. I have always believed that there are more varieties of Alzheimer's disease than we recognized. This research confirms my suspicions. If you're baffled by amyloid plaques and microtubule tangles, there are some links in the show notes below. And of course, we explain what we know about them in Beating the Dementia Monster. There's also a link to our Alzheimer's disease blog, where we have more than 550 articles on my experience with my disease and the fascinating things I've learned on my journey. So thanks for watching. I hope that you find what we do here interesting and you'll want to come back. Writing comments, liking, sharing, and subscribing all encourage YouTube to promote our channel. Have a great day.